Okay, we're good to start. And hello, everyone, and thank you for joining our webinar today. We also appreciate you submitting your questions in advance, as it helps us tailor the session to focus on what matters most to you. My name is Gabriel Escalante, and I will be your moderator for today's session. During this webinar, Tristan Fromager, application specialist at Vilber, will demonstrate how modern imaging tools can improve data quality, reproducibility, and quantification in Western blot. He will also discuss strategies to optimize your Western blot workflow, along with alternative detection methods and their associated benefits. Just before we begin, a few housekeeping notes. If you have any questions during the session, please submit them through the Q&A panel, and we will address them at the end of the presentation. If we, by any chance, are unable to answer all questions due to time constraint, we will follow up afterwards. A Q&A document and a recording of the webinar will be posted on the webpage, and I will send you the link once they become available. Without further ado, I will now hand over the presentation to Tristan.
Hello, everyone. Thank you to be here for this webinar. Thank you for joining this exclusive webinar in partnership with Scintica. So today we will talk about how to succeed in Western blot. So as Gabriel said, I'm Tristan Fromager, and I work for the company Vilber as an application specialist and molecular imaging expert. So Vilber is a French and German manufacturer of high-end imaging system for molecular biology research. And indeed, we design a system for Western blot and gel imagers, but also for advanced in vivo imaging system for preclinical research. So here is what we will cover during today's sessions. The idea is to provide you with practical tips, okay, on how to optimize your Western blot and to help you to choose between chemiluminescence and epifluorescence imaging techniques. So let's start with the Western blot protocol. And first the sample preparation and the cell lysis. So if you are working on cytoplasmic proteins, a milk detergent like Triton X unread works well, okay? But it's very important also to add protease and phosphatase inhibitor. Otherwise, your protein might degrade before you even run the gel, okay? After lysis, can centrifuge at 4 degrees for about 15 or 20 minutes, and this step separate the solid protein from the cellular debris and carefully collect the supernatant and keep it on the ice. Okay. Next, you can add a loading buffer. This loading buffer contains SDS, and this SDS gives protein a uniform negative charge, and you have also a beta mercaptol on this buffer, and the beta mercaptol breaks disulfide bonds, so protein are fully denaturated. And then we can eat at 95 degrees for 5 minutes. But if you have sensitive protein, you can eat at 70 degrees. It will be safer. After that, you have to separate, okay, your protein with electrophoresis gel. The percentage of polyacrylamide is critical, okay? For small protein, you can use 10 or 12% of polyacrylamide for your gel. And for larger proteins, you can use 6 to 8%. It works better. Choosing the right gel percentage ensure a good hand separation because it's the most important thing here. For the electrophoresis, It's typically run at constant voltage, okay, around 120 to 150 volts. And you will stop the migration until the dye front reach the bottom of the gel, okay? Uh, there is no exact time, to be honest, but it's around 40 minutes. It depends of the size, the gel size, the system, and also of your protein. After the migration, you have to transfer your protein. This step is critical, okay, because poor transfer means poor detection. So there are two main approaches, and we will see that today. There is the wet transfer and semi-dry transfer. Uh, each has advantages, okay, depending on your protein size and experimental properties. The first transfer is the wet membrane transfer. So the gel and the membrane are assembled in a cassette and fully immersed in a transfer buffer. The standard buffer contains trace, glycine, and about 20% methanol. Okay. Um, it's a long technique, okay, often 1 hour or more, but it's very, very efficient, especially for large protein. So if you are working with high molecular and weight protein, wet transfer will be, according to my opinion, okay, the safest option. And the second technique is the semi-dry membrane transfer. It's faster than the wet transfer. The gel membrane sandwich is placed between electrodes and yes, between electrode plates soaked in a transfer buffer. It can reduce the transfer time significantly, as I said. However, there is a risk of membrane drying, which may the transfer. Okay, this way affect the transfer. Um, these kind of techniques work well for routine, okay, for routine application and for small to medium protein. But as I said, for high protein, it's better to use the wet techniques. After transfer, we block the membrane. It's very, very important. It's an important step. This step prevent non-specific antibody binding. Okay, typically you have two options. Okay, you can use 5% milk in TBST. Okay, or 1% BSA. But to be honest, milk is economical and work well in many, many cases. Um, and BSA can be used for phosphorylated proteins. But the most important thing is to block the membrane to reduce background and improve specificity. And the last step is the antibody incubation. So first, we dilute the primary antibody, okay, in the blocking solution to a concentration of 25 picogram per millimeter of antibody. And then we incubate the membrane with this primary antibody. And you can incubate your membrane during 1 or 2 hours at room temperature or overnight at 4 degrees Celsius. After incubation, you have to wash the membrane several times with a buffer. Usually we use PBS, okay? It's very important to wash because you have to remove any excess of primary antibody and that reduce the non-specific binding, okay? And after, you have to prepare the secondary antibody by diluting it in a blocking solution to a concentration of, yes, 2.5 picogram per millimeter. And finally, you have to incubate the membrane with the secondary antibody for 1 or 2 hours at room temperature. And at this moment, the second antibody will bind the primary antibody and allow you to detect your protein of interest. If you want my opinion, it's better to incubate for the primary antibody. It's better to incubate the first antibody overnight at 4 degrees Celsius. It will be better for you. Now we will move to chemiluminescence troubleshooting. And the first troubleshooting is the weak signal of the protein of interest. So the aperture of your camera control how much light reaches the camera sensor, and a small aperture which corresponds to a high F number. Okay, we'll see that, but for example, the number is close to 1 or 0.95, and it's a high F number. So when you have a high F number, lets in less light, which can make the image darker if the exposure is not adjusted. So, as I said, the aperture control how much light reaches the camera sensor, and our system, the Fusion Absolute, has the largest aperture on the market with a value of 0.7. So with our camera, it's possible due to the lens aperture to detect a low quantity of protein. The second troubleshooting is the background noise. Okay, long exposure can increase thermal noise in the image. Okay, a camera produce heat. Okay, and this heat create thermal electron inside the sensor and add unwanted signal. So as a result, a high background can appear and may mask low intensity bands. Okay, and with this background, it will be impossible for you to detect your protein. That's why the camera cooling is very important, because it helps to reduce thermal background noise during image acquisition by keeping the camera at a very, very low temperature, and that can minimize thermal artifact and get a cleaner images. And with the Fusion Absolute, it's possible to cool the camera down to minus 90 Celsius degrees. In chemiluminescence, the third troubleshooting is saturated band. Okay, saturated bands happens when a pixel exceeds its full well capacity. Okay, for example, it's gray level. Okay, so you have the scale is between 0 to 65,000 gray level. So a larger pixels can collect more signal before reaching saturation, which help preserve image details. So the number of pixels is important, okay, for getting a high definition image. However, in scientific imaging, the pixel size it's the pixel size that's really matter for maximum sensitivity, and that allows us to collect the most quantitative data. And the pixel size of the Fusion Absolute is large, okay, very, very large. It's 8.6 micrometer per square. And normally, if you look at normal camera, it is around 3 micrometer. So with our camera, it's possible to detect low quantity and high quantity of protein, but without saturation. Okay.
Well, thank you, Tristan, for the presentation so far. We will take a brief moment just to launch a quick poll to the audience, and the questions are going to be the following.
Okay.
So, the first question is going to be, do you currently use Western Blot? Answer is yes or no, depending on your choice. The second question is going to be what proteins and or pathways are you planning to investigate, if any? And what issues, obstacles are you currently facing in your experiments, if there's any, again? Please feel free to answer these questions. We're going to take a couple seconds, minutes, maybe. And as I say this, I just want to highly encourage again that in the Q&A section, you can ask any outstanding questions that you have or any specific question that you might want to address either with Tristan or with my colleague Jade. So we're just going to wait a couple seconds.
I think there is a lot of issue when you experiments with Western blot. It's normal.
Yeah, I've heard it's a lot of manual work that needs to be done. So
Yeah.
I
Yeah.
I understand. Okay, well, I see people answering, and I see people also asking questions in the Q&A section. Thank you so much. We appreciate it.
Thank you.
Okay, we're going to take a couple seconds more. Okay, I'm going to end the poll. Thank you for the answers. And I will now hand over the presentation back to Tristan.
Yeah.
Oh, apologies. There seems to be some issues with our Wi-Fi. Uh,
Uh,
s- oh, there we go. You're good. You're good now, Tristan. We see you.
you see me? Okay, sorry.
Yeah.
You want to access to the share, right?
Yeah, you can continue with the presentation.
Okay. Okay. Perfect. Okay. So yes, okay. So let's move to epifluorescence now. So the epifluorescence techniques. So epifluorescence imaging is a technique where light shines onto a sample and the same lens collects the light, okay, emitted by fluorescence labels. Special filter separate the excitation light from the emitted fluorescence from the dye, so that that's only the signal from the Western blot is detected. Okay. For Western blot imaging, epifluorescence provides a wide dynamic range, okay, a stable signal over time, and the ability to detect multiple targets at once. And chemiluminescence, on the other hand, is more sensitive, okay, and also cost effective because you just have to purchase ECL, for example. But the signal is transient and multiplexing is impossible with chemiluminescence. And now we will see epifluorescence troubleshootings. And we will start with the channel crosstalk. The trouble the fir- This occur when the excitation wavelength or emission of one dye is too close to another, causing signal to overlap, okay? And to avoid this, you have to use spectral filtered LEDs that precisely match the excitation peak of your selected dye. And you have also you also have to use high quality narrow band pass excitation and emission filter. And with this kind of filter and excitation, you will minimize spectral overlap between channel. Okay. So it's very important, yes, to understand that. If you have a crosstalk channel, it's due to your imager, it's not due to the die, okay? The second troubleshooting is weak signal, the one for chemiluminescence. But here, it's due to the LED. Okay, you need a strong emission, and by using a high intensity pulse LED, it will improve the excitation efficiency and give you a stronger fluorescence signal. So yes, to avoid this issue, you have two main strategy. So thanks to our spectra channel system, first you reduce crosstalk, okay, because you will have precise excitation of the target, and secondly you will enhance weak signal detection with pulsed LED, and pulsed LED allow detection of low intensity fluorescence. And also, thanks to filtered excitation through the spectral channel, only the specific wavelength for your dye is delivered, okay? Improving specificity and overall image quality, giving you more accurate and reproducible fluorescence measurements. The third troubleshooting in fluorescence is the high background noise, okay? By using large band filter, you will capture unwanted wavelengths, which increase the background because you will capture also the excitation of your module. So to avoid that, Fusion Absolute filters help reduce background noise by allowing only the specific emission wavelength to reach the detector. They increase the signal to noise ratio by blocking unwanted light from other source that I said from the excitation. So yes, very important to look at your filter and about their blocking range. I think it's the most important troubleshooting here is the non-uniform light, okay? It's due to fixed LED because fixed LED creates a bright central halo, which cause uneven light intensity across the sample. As a result, some area may be under or overexposed, reducing image quality. And fixed LED are therefore limited for applications that require uniformification across the entire field. For example, if you have a big membrane, it will be impossible to have a uniform light. So sometimes you don't detect your protein at the corner, for example, of your membrane. And to avoid that, and on this slide also you can see we have developed a new spectral excitation called absolute illumination. Okay, the idea is quite simple. Okay, the light source ensures that the entire sample receives exactly the same amount of light, and this helps eliminate intensity variations that could otherwise affect the accuracy of the analysis. In practice, okay, the whole blot is evenly illuminated from the center to the edge of your membrane. And as a result, you get a truly homogeneous signal, which significantly improve both reliability and reproducibility. And now we will talk about the interest of photon calibrated optics. So first, what is the NIST calibration? NIST, which stands for the National Institute of Standard and Technology. It's an official organization, okay, that provides certified physical standards. And for the fusion, for absolute quantification, so we will see that also a little bit after. But we rely on calibrated light standards, and this standard emit a precise and known number of photons per second. For example, we use a reference that can emit exactly, for example, 80,000 photons per second, and answer accurate and reproducible measurements. The interest of the fusion of the absolute quantification is to convert counts, so counts it's like gray level, okay, into phantom flux. We image NIST calibrated reference, as I said, under controlled condition like exposure time, binning, aperture, and also distance, okay, so the field of view. And we subtract the dark current and any background light to ensure that only the true signal is measured. And finally, we create a calibration curve, allowing us to translate camera counts, so as I said, gray level, to real photon flow values. Okay. The calculation method is very simple. Okay. It's identical to what we use for gray level. Okay. So except that here we are working with an absolute unit measurement. So instead of relative intensity value like the gray level, the results are expressed in real physical units such as photon per second, and this value enabling true quantification. It's very important to understand that, because, for example, if you change the exposure time with gray level, you will change also the value. But here, with photon flow, the value is the same. So it's possible to, at different exposure time, to quantify a blot. But of course, if you prefer to use the previous method, like with the gray level, it's also possible with our system, but we provide also this tool. So how to conclude, how to choose the right blot imager. First, as I said, the calibration is critical, okay? With National Institute of Standards and Technology calibration, you move from an arbitrary gray level to true absolute quantification. The second is the sensitivity. Okay, it's very important to detect low quantity of protein, and a large aperture is important. A cooling down to minus 90 degrees Celsius, as I said, is also very important. But the most important thing is the large pixel size. Because it will be able with these three parameters to detect low quantity of protein, but also in the same blood to detect high and low quantity without saturation. So for your quantification, it's very important to have big pixels and a large aperture and a great cooling now. And third, for the fluorescence, okay, the illumination source is essential. It's very important to have a uniform light source, okay, as I said. And also, it's very important to have narrow band pass filters to select precisely the emission light. And finally, I think the most important thing is to have a all-in-one system. So it will be possible for you to, if you need sensitivity to take picture in chemiluminescence, and if you want to do multiplexing, it will be possible with fluorescent channel. And also with our system, it's possible to take a picture in with gels, for example, with a UV pad or a blue pad. And also we have a white pad. So, I think you have a lot of questions.
Yeah, perfect. Thank you so much, Tristan, for the excellent presentation. As mentioned before, we're going to move on to the Q&A session, and Dr. Jane Merrill will be assisting us with addressing some of the questions. So we have a couple more questions over here. And again, I encourage everyone to ask us any questions right now, or we can address them after the session. But the first question is the following. What do you need to change in the sample preparation if you deal with membrane proteins?
For example, for membrane proteins, wait, I will go to the slide. It will be easier to understand. Um, so for the sample preparation, for cytoplasmic, as I said, you have Triton X1, you have to use Triton X100. For membrane protein, it's more difficult, to be honest, and it depends of your protein. So if you want, you can send me information about your study, and I can give you some advice about specific proteins. Because for cytoplasmic protein is very easy, but for membrane protein it's more difficult and it depends on many parameters. So yes, I prefer to search before to give you some advice.
Okay, perfect. Thank you so much, Tristan. We have another question, and for
Yeah.
this one, Jade can help us out. Even maybe Jade, is it okay if you go over it? Because there's a lot
Yeah,
of scientific terms I don't want to get wrong. So
yeah,
I'll let you handle it then.
you bet. Thanks, Gabe. Okay, so the next question is essentially saying, for some of my proteins, when I detect the phosphoproteins, they're fine. So think phospho AKT, phospho AMPK, phosphostat 3. And then after stripping the nitrocellulose membrane and incubating the total ones, I end up with faded bands for the total proteins. So this person is going to be using 15 micrograms of protein, often using pre-made gels of about 20%, with an 18 well comb. So relatively narrow wells. Sometimes the proteins also become stubborn to strip, even though they were removed before. These uncertainties are going to cause batch-to-batch variability for us, and so it's a little bit unreliable. How can I go about solving some of these issues?
I think it's the problem is due to the transfer. If you have weak bands, it's due to the transfer. So maybe you can use wet transfer. I don't know if you use that. So if you have faint bands, maybe it's good to move to the wet transfer. And if not, maybe your imager is not enough sensitive. So you have to move to another system with a biggest aperture, I think. And maybe with your system, you can use binning if you want to increase the sensitivity and detect faint bands.
Yeah. Some from my own personal experience too, sometimes if you try lowering the percentage of your gel, for the proteins that you mentioned, so AKT, STAT3, and PK, based on sort of their molecular weight, sometimes they can get trapped in a 20% gel as well. So if you try lowering it to maybe 10%, seeing if that helps to pull them out more. Um, with the membranes as well, using a nitrocellulose, I'd be curious to know if you've tried doing PVDF, and seeing if that helps at all. And then sometimes too, if you are wanting to load a little bit more protein and maybe consider using wider wells, sometimes that can help to pull out more of the protein. So with
Yes.
that, Gabe, oh, sorry, Tristan, did you
Yeah,
want to
no,
comment?
no, continue.
No, I was gonna pass over the Q&A moderating
No.
back to Gabe, but if you wanted to make any kind of comments to that, please feel free.
No, it's just about nitrocellulose and PVDF. It's just, for example, in fluorescence, it's better to use PVDF because you have a lot of photofluorescence. It's just to make an advice, just like that. It's okay.
Awesome. All right, Gabe, I'll pass it back to you. I think there were some other questions that were submitted whenever registrants were doing the registration. So feel free to moderate those.
Okay, thank you so much, Jade and Tristan. So the next question we have, how can I optimize my Western blot to apply, whoa, my apologies, to obtain clean, low background, non-saturated results?
Okay. There's two options. Okay. The first is to properly block your membrane. Okay. With the blocking solution, as I said, with milk or other blocking solution. And also, you have to look at your imager, and maybe the cooling is not enough. So you will need a better cooling if you want to reduce the background and just to have a clean membrane.
Perfect. Thank you so much. The next question is, how can I ensure reliable quantification and reproducibility across experiments while staying within the linear detection range?
Yes. Yes, for that it's linked to the calibration. Okay, as I said, with a nice calibrated system, it's possible to link gray level to a scientific and reliable value. So we call that photon flow. It's photon per second. So with the absolute quantification, it's possible to ensure a reliable quantification and it will be possible also to obtain the same value with different exposure time.
Thank you. And the next question is, how can I adjust imaging parameters to accurately capture both strong and weak signals without losing information or introducing bias?
Um, yes, okay. You have two options, but for example, if you change the binning of the camera, so you will merge pixels, um, you will lose information and you will introduce bias also. So if you need if you want my opinion, it's important to have great pixel size in the native resolution because there is no bias and there is no it's impossible to lose information because it's the native resolution. So, to respond to your question, it's possible to use the binning, but it's not the best way. So the best way is to use a system with big pixels, a big pixel size at the native resolution.
Thank you again, Tristan. We're going to wait a couple moments in case someone's typing a question. And if not, we're going to move forward and end the session. Oh,
Okay.
we have two more questions. Okay.
Oh, perfect.
Jade, I might need your help with reading another question. It's very scientific, and I'm not sure if I'm going to get the right pronunciation with it. Would that be okay?
Yeah, no problem. Do you want to type in, or sorry, speak out the one that's a bit shorter first so I can get a chance to read through the longer one just in the
Sure,
background? Sure.
that works. Okay, so the next question is validating and optimizing primary antibodies and blocking agents conditions. Um, I'm I'm not sure if
I'm
if
not sure
there.
to understand the question.
Yeah, if you could just clarify in the Q&A section, gents, we would appreciate it. And for now, we're gonna move on to the next question, if that's okay, Jade.
Yep, no problem. So this person is asking, or they're providing some context. So we had trouble with primary HRP coupled antibodies staining every band, including the markers, in the recommended working concentration from the company, so in the data sheet. diluting the antibody up to 30 times and to up to 100 times more than the recommended seemed to help a little bit. But whether it worked was highly dependent on the sample that we were dealing with. So either a purified protein or a cell lysate. Is this a common problem and how can we deal with it?
Hmm. If you have a problem with the primary antibody, you have to change it. To be honest, it's the only solution. Maybe you can block your membrane during maybe 1 or 2 hour in blocking buffer. But yes, if you have a problem with if it's not specific, if your primary antibody is not specific, you have to find a solution and to find another primary antibody.
Thank you, Tristan, and thank you, Jade. We're going to move on to the next question, which is thank you, gents, for the submission. So it would be approaches to validating and optimizing primary antibodies and blocking agents slash conditions.
Could you repeat the question?
What I guess it would be what are the approaches to validating and optimizing primary
Uh,
antibodies and blocking agents slash conditions?
as I said, for the antibody incubation, it's better to incubate it overnight at 4 degrees. This is the best condition for me to incubate primary antibody.
Okay, perfect.
Do you
Okay,
have any, sorry to interrupt you, Gabe. Tristan, do you have any recommendations on doing the validation process and other ways of optimizing? So going outside of doing the kind of overnight incubation, are there other sort of trial and error tips that you can come up with that would help to essentially validate the antibodies, or is it just a matter of kind of doing day after day after day of trying something different because again,
Yeah,
there are so many manual steps that you can change things with. And then of
yeah.
course to see the impact does take time.
This is the problem with Western blot. I present you a protocol, okay? But it depends on your protein. If it's a specific protein or uh, there is no I don't know how to say, but the protocol can change every time. If you have a big protein or a small protein, it's a phosphorylated protein. So yes, it's very difficult to make these kind of things.
One thing that I would say from again my own experience is sometimes whenever you're working and hitting the same problem over and over, and you feel like you've tried kind of everything and changed up every little step, and you're still not seeing success with it, sometimes it's helpful to have a lab mate to actually spend the day with you and watch what you're doing, because it could be something very small that is affecting the outcome and keeping you from getting your data properly. Again, also having conversations with your PI and kind of gathering their, expert ears, working with Western Blot to see what they might be able to pick up on. Because again, sometimes it's something very small and nuanced that we're doing that's keeping us from reaching what we want.
Yes, and to be honest, also the most important part in the Western blot process is the last the last one. It's very important, as I said, to have a very sensitive camera because you can succeed every step of your Western blot, but if your system is not sensitive enough, you will detect nothing. So yes, very important to look at the imager.
thank you, Tristan and Jade. Appreciate the responses. So, we have now come to the end of today's webinar. I would first like to thank Tristan for the excellent presentation, Jade for helping us in the Q&A section, and the viewers, of course, for attending to this session. I hope this session has provided you with a clear understanding of how to optimize your Western blot workflow and improve the quality of your imaging data. If you have any additional questions in the coming days or weeks regarding the modalities we discussed, please feel free to contact us at Scintica, and we will be more than happy to continue the conversation. Thank you all for taking the time to join us today, and we look forward to seeing you at a future Scintica event. Have a great day.
Thank you.